Hi, everyone. Welcome to the progress reports on the rising stars in chemical ecology. About 20 months ago, specifically on August 9, 2022, we organized a symposium named Frontiers in Chemical Ecology. And we wanted to democratize chemical ecology, so we offered that symposium as an open online for everyone who wanted to attend. And thanks to the sponsorship from Biochemistry Bio, also known as Biochemistry Research Incorporation, we are able to put that inside of the joint meeting of the International Society of Chemical Ecology and the Asia-Pacific Association of Chemical Ecologists. That meeting was organized by my friend Alvin here, and Alvin was very open-minded to that idea, and Biochemistry decided to sponsor that. So at that meeting, which was the 37th annual meeting of the International Society of Chemical Ecology and the 11th meeting of the Asia-Pacific Association of Chemical Ecologists, we had this symposium with the stars in chemical ecology, rising stars in chemical ecology. Of course, there are many rising stars in chemical ecology. We could not have a symposium because it would take days or perhaps weeks to have all the speakers. But we line up with a number of speakers. And after that, many people asked me, what happened with it then, from then and now? So this is the progress report to share some good news with you, what they have been doing, what accomplishment was done. You have to understand that some people were there still as a postdoc at that time. So it takes time for the lab to build up everything. So that's take time, and sometimes not much progress can be achieved in 20 months. 20 months in terms of research activity is not a very long period of time. But before we start, I want to explain the T-shirt. Okay, this T-shirt is in my possession for 28 years, and I'm wearing that for the first time today because many of you know me well and know that I do not wear T-shirts when I talk in public. I do not wear T-shirts in general because I have very weak skins, unlike Jerry McNeil that only wears T-shirts and comes to meetings, talks in T-shirts. I never go to meetings in T-shirt, and this is a formal meeting, but I decided to wear this one because I have this T-shirt from the 13th annual meeting of the International Society of Chemical Ecology. And where was that meeting? In the Czech Republic, in Prague, exactly the same place where the International Society of Chemical Ecology is going to convene this year for the 39th annual meeting of the International Society of Chemical Ecology in Czechoslovakia, in Prague. If you wanted to attend, and I hope you will do, please look at this QR code here and scan that because we are going to have information, the link direct to the annual meeting of the International Society of Chemical Ecology. I can guarantee that's going to be a wonderful meeting because we are not going to be able to bring the same people that were there. But remember that in there, we had Kenji Mori gave the social lecture, the late Professor Kenji Mori. And that time also he got this, the silver medal of the International Society of Chemical Ecology. In that particular year, he shared the medal with Kenji Mori. Not they shared the medal in half, but they made that year 2 silver medals because there were so wonderful candidates, they could not select the only one. So they gave one to Kenji Mori and other to Jack Pastilles. At that time, Louise Vett gave the social, not the social lecture, the Silver Star Simeon lecture. Kenji Mori gave the social lecture, what was sensational. I remember Thomas Hartman talking about the insect-plant interactions. And also the legendary Mary Blanc was there, and he spoke after me. One day I will have an opportunity to tell you what he told you about me just after my presentation. So it's something interesting, and maybe I should tell it now. Okay, so he said that I want to speak as fast as Walter Leal, but my wife, her name is Donna, my wife highly recommend that I use verbs, prepositions, and everything. So I can't speak as fast as Walter Leal. So That was Mary Bland that we love so much and we miss Mary Bland so much. But speaking of people that we miss so much, I'm going to get the great pleasure of presenting in Prague this time the Vitico Franks Dax Chemical Memorial Lecture to honor Professor Gerald Meinwald. It's going to be such a great pleasure to have this opportunity, and we are going to be convening in Czechoslovakia in Prague, again, July 14 to 18, 2024, and I hope you can attend. So without much further ado, I already talked too much. Let's get a progressive report on the rising stars in chemical ecology. And I'm going to start in here in alphabetical order, at least for the first one. Ali Afif, he was here 20 months ago as a postdoc from Johns Hopkins, and he was presenting and announced that he is going to have a new lab, a new position as assistant professor at the Drexel University. Ali, what happened between then and now? Can you give us an update?
Thanks for asking, Walter. I think it has been over a year and a half now. I can't believe it just flew that fast. So during that time, I started my position at Drexel, where I started my lab. It's a really small lab now. There are 3 additional lab members in my lab. We are currently working on 2 main projects. One is on oviposition of Anopheles mosquitoes. So mainly, we would like to identify cues, attractants, or repellents for egg laying in Anopheles mosquitoes. And mainly, we are really testing potential cues that work for other mosquito species, such as Aedes and Culex, with Anopheles mosquitoes. So when we started using some of these cues, we found that 2 repellents, one is called butyl anthranilate, that works as a repellent for Aedes aegypti mosquitoes, that actually work also as a repellent for Anopheles mosquitoes at a broad range of concentrations. But at a very low concentration, it works as an attractant for Anopheles mosquitoes. We also found that another repellent for Aedes aegypti called ethyl anthranilate, interestingly, it was working as an attractant for Anopheles mosquitoes at different concentrations. And this actually suggests that chemicals don't work the same way for all mosquito species, and there are differences of how these mosquito species respond to different chemicals or use or odorants. So this ethyl anthranilate, in addition to being attractant for Anopheles mosquitoes, it also kills eggs. So mainly mosquitoes lay their eggs on ethyl anthranilate, and then the eggs never hatch. We don't really know why this happened, why mosquito would take a decision to lay eggs on a substrate that would kill their eggs. But it's really interesting that we can use that chemical in traps in order to attract mosquitoes to lay their eggs and also kill them without use of additional insecticides. So we are continuing to identify more chemicals that work as attractants or repellents for Anopheles mosquitoes that we can use in traps to attract those mosquitoes and kill their eggs. In addition, we would like to also identify receptors, odor receptors that are used by Anopheles mosquitoes to sense those chemicals using a method that we developed during my postdoc at Hopkins at the Bottle Lab, mainly by using calcium imaging in Anopheles mosquito antenna, followed by RNA-seq of tissue that contain the neurons that respond to the certain chemicals. So this is the first project that we have been working on. The second project is a collaboration with Megan Creighton, which is a professor at Drexel's biological engineering department, and we are preparing a chemical called hexagonal boron nitride, which is a skin-safe chemical widely used in cosmetics, and it can be exfoliated into 2-dimensional platelets with nanometer scales thickness. This chemical, we propose that it can work against mosquito biting through two mechanisms. One, that it can decrease the evaporation of skin odorants when we apply it on the skin, similar to what DEET does. So previously, I found that DEET mask the evaporation of skin odorants from the skin, which also decrease how mosquitoes are attracted to us when we use DEET. So we think that HBN, hexagonal boron nitride, works the same way by masking our human skin odorants. And we did some earlier experiments, and we found through hand-in-cage assay that applying that chemical on the skin would prevent mosquito landing on the skin, which means that it's either working as a true repellent that can directly repel mosquitoes through their smell, or that it works as a masker like DEET. So this is what we are going to do next. We will find out if it actually works as a true repellent or as a masker. We will also test how it physically frustrates mosquito biting and penetrating the skin. And this is a second mechanism that we think HBN works with. So it can physically frustrate the biting mechanism itself. So this is a more of a long-term project that we will be working on. So hopefully the results that we got from the oviposition project and the HBN project will be published soon, hopefully by the end of the year. So these are the first preliminary results that we are getting out soon.
Ali, that's great progress. I appreciate how much you have done, and I'm very interested in these oviposition attractants, and you focus on your research on finding chemicals that might be useful in terms of control mosquitoes. And I'm so intrigued by these chemicals that you found now that kills the eggs, not only attract the females, they lay the eggs and you kill, because one of the things that we have to do in chemical ecology is to combine an attractant with a toxic agent, such as BTI, but looks like you found the two bullets in a single one. Can you explain that, elaborate more on that?
So yeah, that was actually surprising and interesting because we don't really know why a chemical would attract mosquitoes to lay their eggs on, but then it actually, those eggs never hatch. So we found that it's like really at very high concentration, it's attractive. And also at 100 ppm concentration, it is attractive. But we don't really know why mosquito would choose to actually lay eggs on something that would kill their eggs. It's just interesting. I think it would work well for really attracting mosquitoes and killing their eggs. But we don't really know how mosquitoes evolved to really get attracted to this chemical while it's lethal to the eggs. Maybe it resembles some smell that is not lethal to the mosquitoes. But yeah, we don't know for now, at least.
Thank you. Thank you very much. Since we are talking about mosquitoes, everyone wants to repel the mosquito. And what a wonderful idea to wear soap, to take a shower and be able to repel mosquitoes the next day, or the day that you take a shower with such a soap. And the Clloé Lahondère, she's work on this very topic, and she's going to give us an update on what's going on in her research activities. Clloé.
Hello, Walter. Yeah, things have been going well here. You might remember that we presented work about the effect of soap application on mosquito-host interactions during the meeting in the summer 2022. And we were able to publish that paper in the spring of 2023 in iScience. And so just like a quick reminder, we found that when people apply soap, so they wash with soap, it basically changes their olfactory signature, so the way they smell. And mosquitoes are gonna be affected by that. So they sometimes, depending on the type of soap that people use, they might be more attracted to the host or more like repelled. So it's really dependent on the type of soap that people use. But also it's like a soap and person combination, which is important to think about. And we also found that there are some specific chemical composition that are driving this, and that we can use that knowledge for creating artificial mixtures. So we were able to create an attractive mixture and a repellent mixture by using this knowledge. So that was pretty exciting. And this actually created a lot of, got like a lot of attraction from the media. So we had about 40 radio and TV interviews and a lot of press articles. And I just give you a couple of examples here. So we had like an interview with the CBC radio. We also had a very nice article. It's in French, but it's in Le Monde. And another example was a New Scientist article. And so it's great because this is really what we wanted to have, is like translate that knowledge and make sure that it's also available for people. So now we are expanding that work using more volunteers and more soap. And so we hope to have like more data in the future and be able also to make predictions based on, for example, formulations of soap, and also develop potential new repellents and attractants. So this is my main update, and yeah, thanks so much.
Thank you very much. That is good to know. And can you elaborate how people are going to be able in the future to use a certain brand of soap to not need to, for example, to use DEET or other repellents?
That's a very good question. So this is something that we are exploring now because we found that there are a lot of effects that are driven by the host itself and by the soap that they use, and that those effects might not be the same depending on the type of host and also the soap. So we are exploring those questions, see if we can develop any new formulation of soap that could be more like drive that repellency that we noticed for some of them. Like one of the compound of interest was linked to coconut derivatives. So this is for us like a thing that we are exploring right now. So, stay tuned. Hopefully in the future we'll be able to chat a little bit more about this data.
Thank you very much. Chemical ecology is a fascinating area. There are so many branches of chemical ecology, but I like to see what's going on in Greg Pask's lab. And for that, we are going to ask Greg Pask to give us an update from his position at Middlebury College. Greg.
Thanks, Walter. Yeah, it's really great to be back and hear about the rising stars in Chemical Ecology group, hear what everybody's been up to. We've been busy kind of starting some new avenues in insect chemical ecology, looking into unlighted firefly species, namely the winter firefly, Phytinus coruscus. It's a very unusual firefly that it doesn't have this big flashy light organ that we recognize in summer evenings, but instead they're diurnal and they've lost the ability to produce light as adults. So it's long been thought that they are out using kind of the ancestral form of mating communication, which is pheromone communication, but nobody's identified the pheromone yet until my collaborators, Sarah Lauer and Doug Collins at Bucknell University and Jocelyn Miller at UC Riverside got together, and our research team went and kind of identified the pheromone, the sex pheromone for this unlighted firefly species. And since then, we were able to kind of do a lot of classic kind of chemical ecology techniques of GC-EAD to kind of identify that there was a hit in this extract. Jocelyn and his students and postdocs kind of went together and identified this compound and then synthesized it for us. We were able to kind of go out and into the field with a kind of a very affordable bioassay system and see if this individual compound, this hydroxycamphor molecule, could actually attract these fireflies, males in the field. And we did that, and after just 24 hours, we found tons of male fireflies on our pheromone baited traps and nothing on our solvent traps. So the stats were very nice and easy for that. And then since then, we went ahead and tried to look at the antenna, see how they're starting to detect this molecule. So we went in, used some scanning electron microscopy to characterize potential chemosensory hairs on the antenna. And then we got to do what we do best, and what we love to do is stick electrodes into those hairs, do some single syncyllum recording, and actually identified a specific hair, a basiconic type syncyllum on the antenna that responds to this pheromone. So now we know kind of, yeah, hey, here's this pheromone that the species has been using, and now we know what part and what region of the antenna that they're using to detect it. Right now, I'm actually on leave for my pre-tenure leave. I'll be going up for tenure and promotion in the spring 25 semester. But right now, because I'm not teaching currently, I'm able to kind of dive into a lot of research and actually try my hand at doing a lot of insect chemical ecology research in the field. So we're kind of using the pheromone that we now have to start to understand more aspects of the natural history of the species, because most insect species, once they become adult, they are very motivated to start reproducing. But this winter firefly species actually comes out in the fall and then rides out the winter as an adult, undergoing diapause, and then comes out, and then there's this kind of little bit of a delay until when they become reproductively active. So we're trying to use the pheromone to kind of have a lot of insights into that progression to being reproductively active, and then getting into, you know, all sorts of other future questions now that we have this pheromone component. So yeah, I'm excited to hear what everybody else has to say, and happy to take any questions.
Excellent job. I'm so glad that you published that work in the Journal of Chemical Ecology. We are very proud of the journal to be able to attract you to publish such an interesting paper. I'm sure that I'm not the only one who loved that paper. Maybe I can ask Nicoletta Faraone. Nicoletta, have you read that paper? What do you think about?
Oh, yes, I read the paper, Greg, and it was very, very nice. Actually, I can tell you that I reviewed that paper, and I really enjoyed the reading. It was fantastic, very well written. And I found also, as a chemist, I appreciate very much the synthesis of the hydroxycamphor. Now, I was wondering, those winter fireflies, are they carnivores? Are they, what do they eat?
That's an excellent question, Nicoletta. Thank you. And thank you for reviewing the paper. So yeah, the winter fireflies, like most fireflies, are carnivores in the larval stage. They're eating slugs, snails, earthworms, and they really build up a lot of energy stores, and they're like little tanks moving around the soil. And then once they become adults after pupation, a lot of your lighted fireflies that you see out in the summers, they might not be eating at all. It's not believed that they might even have functional digestive systems like some other insects that we know like that. But the winter fireflies, because they have such a long duration from adult to actually being reproductively active, people find them in maple sap buckets and potentially eating some pollen from certain plants, but it's still an open question of how they're keeping that energy up during the reproductive season.
I also, because I was interested about the structure of this pheromone, and it's a terpenoid. So I was trying to think where they might use the substrate and to build up this chemical. So what would be the biochemical pathway? I don't know if it's known that.
No, and like closely related click beetles in the Elateroideae, they also use terpenoids for their sex pheromones. So we have some some umbrella chemical functional groups and nomenclature there, and we're kind of interested to see how they're making it as we move forward. And also, as we all know, that I like what's detecting it.
Yeah, very good. I really enjoy your paper. Yeah, thank you.
Thank you.
I didn't know when I asked that question. I'd ask Nicoletta to ask you the question. I was not planning to disclose anything that was supposed to be confidential, but these days, not as confidential. Some review signing in the
well, it's
report.
published. is published, Walter, so that's okay.
All right. So you didn't want your name to be there if the paper was rejected, correct? All right. So anyhow, Greg, I want to ask you a follow-up question because this is going to be seen by people in many parts of the world, and they know about the tenure process and everything. But there was something that you mentioned in the video that caught my attention. You said that you are going to be on a leave during the tenure. Looks like a mini sabbatical within a tenure period of time. Might be something very special to your university. Could you explain that, elaborate for people, especially those who are not in the United States?
Yeah, so I go up for tenure in about a year, and I have the option of kind of not teaching for either a semester or a full year ahead of going up for tenure. It's kind of time dedicated to research and writing, and maybe writing an additional grant that you hope to have come in before you go up for your tenure review. So I'm doing that right now, and I haven't been teaching at all this semester. It's been very nice, though I do miss seeing more students. And it's allowing me to do really cool things, like I said in the video, of like getting out into the field. I'm not a field biologist by training at all, but these fireflies are pulling me out into the field, and we're pretty fortunate that our field site is actually right down the street from my house. So I can just go out into the field with my students, and then they go back to campus, and I can just go home and cook some dinner.
Speak of tenure, one of the rising star got tenure during this period of time. Between the 20 months that we had that symposium, and now she got a tenure. Nicoletta Faraone, let's get an update from you.
Yes, Walter, thanks for asking. Actually, things are going pretty well, and a couple of important things that happened since last time we talked. Actually, I have a slide that I can share with you, so that summarize the main thing that happened. Well, first of all, I got tenure, so I'm very happy about that. So I'm now officially at Acadia University in the chemistry department. I got two important grants. The first one is through the NSERC Alliance stream. These are grants partially funded by an industrial partner. In this case, I'm working with Atlantic. And also part of the funding comes from the MyTax Accelerate, which is a very good funding stream to support trainees and postdoc and researchers. And for this grant, I'm specifically focusing on the impact of climate change, so temperature and humidity fluctuation on tick response to repellents and acaricide. And then I got another grant, which is kind of slightly different from the things that I usually do, which is the Intentional Initiative Award through Research Nova Scotia in collaboration with my colleague in the biology department, Dr. Susie Curry. So we are looking at studying the behavioral and neuropharmacological activity of psilocybin for exploring the mechanism of psilocybin in promoting, the treatment in promoting mental health and for the treatment of specific mental diseases and so on. And we are using as an animal model. Specifically, I'm working with Drosophila. And my colleague is using fish as a behavioral model and animal model for this. And then I was also invited as a keynote speaker at ChemCon, which is the Atlantic Chemistry Conference. Usually is cover Atlantic Canada. And it will be held at Cape Breton University. So I'm very excited about that. It's a conference specifically oriented for undergrads and will be nice to give a good impact and sharing, my journey between chemistry and biology. And also, I am the symposium organizer for one of the symposia offered at ISCE at the International Society of Chemical Ecology meeting that will be held in Prague this July.
Specifically, the symposium will focus on the chemiosensory system in acrines and other non-insect arthropods. And I'm organizing this in collaboration with my colleague in biology, Dr. Kirk Hillier. So these are my main highlight and the most important things that happen. So thanks for asking.
I'm so glad that you are coming to Chemical Ecology. Once again, congratulations on your tenure.
Thank you.
That's a very well-deserved. People outside don't understand what tenures means. That it means now that you have to work harder because you are going to be asked to participate in more committees and the other things, other functions of the university. But it's much more difficult for the university to fire you if they so desire. They cannot do that easily, right? People say that, there is only two groups of people that has a tenure. Is the Supreme Court, the judge, the justice, and the faculty, right? It's a privilege that we have, and there are reasons for that, so that the university cannot get rid of people easily. Although tenure is not completely bulletproof. Let's realize that. But it's a very outstanding accomplishment, because not only tells that we appreciate what they're doing, but your peers at the university and the higher rank of the university appreciate how much you are doing to the field. And I'm so glad that we are going to bring this knowledge and everything to the International Society of Chemical Ecology in Prague. Can you elaborate a little bit more about your symposium?
Yes, so we put together this symposium with my colleague in biology here at Acadia, Kirk Hillier, focusing specifically on the chemiosensory system of acarines. And also, we decided to expand a little bit to other non-insect arthropods, so to have a wider range of people that might be interested in submitting. We got very fantastic speakers already. And as a keynote speaker, we have in our session Liz Roy, that she's going to talk about poultry mites. So I really hope that some of you will join the meeting. It seems very exciting, the symposia that we have. I think there are about 20 symposia, are really, really exciting. And as you mentioned, Walter, since it's been many years that they haven't been in Prague. This is a big event. It's coming back in Prague of ISCE, so it's I look forward to it, and I hope to see you there.
I'll tell you that one of the best things about this meeting is that I'm going to have an opportunity to meet you in person, because
yes,
I recall that I have met you. I know you well via Zoom and
yes.
things like that, but never met in person. So this is going to be a plus for this meeting. I'm so glad that I'm going to be there. Let's have another update. An update from Trevor Sorrells. He was another one that was a postdoc from Les Vauchal's lab at the time that he was presenting here in this very famous symposium, Frontiers in Chemical Ecology of 2022, inside of the annual meeting of the International Society of Chemical Ecology and the Asia-Pacific Ecology, a joint meeting, and he presented that. So let's hear from Trevor what he has to say about what happened to him and his lab in the meantime. Trevor.
All right. Thank you, Walter Leal, for inviting us to give updates on our labs as part of the Rising Stars in Chemical Ecology Symposium that happened a few years ago. So since the symposium, I have actually started my lab. It's been open for 1.5 years. So the main update I have is that we are in operation now. But we've also received funding from the NIH, the National Institutes of Health, and HHMI, the Howard Hughes Medical Institute. So I was appointed at HHMI a Freeman Hrabowski Scholar, which is awarded to people with the potential to mentor people from diverse backgrounds to do great science. And the Freeman Hrabowski, who is the award is named after, was the president of UMBC, University of Maryland, Baltimore County, for over 30 years. And during this time, he established the Meyerhoff Scholars Program to promote achievement of historically excluded racial groups in STEM fields. And so I take great inspiration from the work of Dr. Hrabowski, and I hope that I can do a part to educate the next generation of scientists. And so to the lab so far, we've recruited 6 lab members, including a lab manager, 2 postdocs, and 3 postgraduate research associates. And so far, our projects are just getting started, but everyone is making really great progress. I'd like to highlight postdoc Joey Zhao, who received a grant from 10x Genomics for a single-cell sequencing project in the mosquito brain. Most of our projects in the lab involve understanding the central brain neurons in the mosquito that control their behavior, especially the attraction to their animal hosts and biting and blood feeding behaviors. And we're also working on understanding the evolutionary origins of blood feeding behavior in mosquitoes, as well as other blood feeding invertebrates. So stay tuned to the Sorrells Lab as we move forward and make progress on these, on our research interests. Hopefully I will have more updates on the science in the future.
Yeah, that's excellent progress. I'm glad that in this period of time, you were able to get grants and build up an excellent team. So the future is bright. So we are going to look forward to read your papers and the success of your students and your collaborators. So having said that, let's shift now, again, going back to Yale, and now with Joel Butterwick. And Joel had this wonderful paper in Nature, and I'm going to ask him what's going on in your lab, although many of you guys already know because it was published in Nature recently. Joel.
Well, yes, thank you for asking, Walter. Actually, we have been busy this past year or so. Actually, we just recently published the first paper from the lab. The first graduate student to join the lab is the first author. And so perhaps I can share a few slides with you to give you a sense of what we've been up to. Yeah, what we had studied was the molecular basis of sugar detection by an insect taste receptor. This was published in Nature in March, and it's actually a really big year for sugar receptors. There's actually 2 other papers to look into. One was published in Science and the other in Cell Reports. But what we had studied is this protein called GR9. It's from the silk moth, Bombyx mori. And what's remarkable about this receptor is that it is only activated by a single type of sugar, D-fructose. And so we want to understand what was the molecular basis for this specificity when even sugars that are very similar in structure don't seem to activate this receptor. And so we use cryo-electron microscopy to determine the 3-dimensional structure of this receptor bound to fructose. And at the top here, I show the cryo-EM density, and the red bits are the extra density that corresponds to the bound sugar. And at the bottom, I show the model that we were able to generate. And if we zoom in on the ligand binding pocket, we can see that there are lots of interactions that seem to be perfectly lined up with fructose. We see lots of hydrogen bonding interactions, we see some hydrophobic interactions, and all of these seem to point exactly to where all the hydroxyls and hydrophobic bits of fructose are. So this is clearly one of the answers for why this is a very selective ligand-gated ion channel, is that the binding pocket seems like it was built to bind fructose. And if we also determined the structure of GR9 in the absence of fructose, and that allowed us to understand what was the activation mechanism. And what we notice is when fructose is sitting in the binding pocket, there's actually a shift in a couple of conserved aromatic residues, a tyrosine and a phenylalanine, which we are calling the aromatic bridge. And this shift in the aromatic residues then allows an interaction between the phenylalanine and the bound fructose. And this shift creates space for the pore helix to then open. And so this is a nice mechanism that allows coupling between the ligand in the binding pocket and the structure of the ion conduction pore, whether it be in a closed or an open state. Now, one thing that was a little surprising, however, is we found that actually lots of other sugars actually do bind the receptor. So we solved the structure of one of these what we're calling non-activating sugars. This is L-sorbose sitting in the ligand binding pocket. And from a first pass, we can see that it makes almost all of the same interactions that fructose did. It makes the same number of hydrogen bonding interactions, a lot of the hydrophobic interactions. But there is one key difference that sorbose lacks, and that's the ability to interact with this aromatic bridge. And so what we see then is in the sorbose-bound structure, the aromatic bridge is still in the closed conformation, and the ion conduction pathway is still closed. And so what that means then is we can think of ligand binding specificity in sort of two aspects of it. One is the coarse ligand binding specificity that's governed by the pocket characteristics. This is clearly going to select for things of the right size, hydrogen bonding potential, a bunch of other things, but it's not really enough to really make the channel selective. And so there's the second sort of fine-tuning mechanism, that's this engagement of this allosteric coupling between the ion conduction pore and the pocket, exemplified here by the aromatic bridge, that provides an additional layer of chemical tuning in this channel. And so here it is, the molecular basis of sugar detection by an insect taste receptor. Here's the QR code if you're interested in checking it out, and I highly suggest it if you're interested in chemical taste, chemoreception, and tuning.
Greg, I'm astonished. This is wonderful science. And what's your thought about that? Do you have any questions?
Well, it's just so great to see that I've been thinking about these receptors, these chemosensory receptors in insects for so long, and then actually, they've been like blobs in my mind, and now that they actually get to be actual macromolecules that we can like dive into. So thank you for that. It's really interesting that there's 4 binding sites here on this homotetramer, and I'm thinking kind of as ORs evolved out of GRs, kind of how do you think, do you think that little pull, that kind of aromatic swing that you saw of that kind of pore lining helix, do you think that's something that we might see when we finally get a heteromeric OR structure?
Yeah, I think that's a great question, Greg. I think there's probably going to be some link like that in all the different structures. Whether it's a pair of aromatic groups or some other link, I really don't know. Certainly, these 2 aromatic groups aren't conserved among most receptors, but for example, in the OR5 from the Vanessa Ruta's lab determined the structure of, there actually are also 2 aromatic groups. And so it might be a little more conserved than we think. I think looking at the generality of this mechanism is something that we're really interested in exploring in the future.
So when you compare the receptor with the only known single standing receptor, that's MHOR5, then you show all these similarities. And can you elaborate for those who didn't read the paper yet? Where is the pore, or where is the ion pore there? Because we talk a lot about that, about the binding pocket and all this interesting phenomena that you discovered now. But can you elaborate a little bit about the pore itself?
Yeah, I think it's something that I always forget to say because I call them taste receptors. And in most systems, that means like a G protein coupled receptor. But for insects, it means a ligand-gated ion channel. And one thing that's interesting about these is, as Greg mentioned, they're tetramers. And so in our case, there's 4 ligand binding pockets, and they surround a central ion conduction pathway. And this pathway is lined by a helix from each subunit. So there's sort of 4 helices that come together. And it's movement of these central helices that then will open the pore and allow ion conduction, and that'll signal to the cell.
So Greg, when you look at your flights, you are going to go after the receptors. What do you expect? What do you expect is that you are going to find quote unquote primitive type with the standalone, or are going to find one that's an orco? I don't know if what's known in your species yet.
Yeah, we have orco identified already, so we expect these to be your typical like orco and tuning OR pairing. As with all beetles, it seems to be the case. I'd like this kind of extra time point that we get when we have these, MHOR5, like the standalone ORXs, sans Orco, is exciting to get another evolutionary pivot point. And maybe insects did it more efficiently with Orco, or maybe not. And there's different trade-offs for each one. So it's exciting to see. Maybe they get less binding opportunities when you swap out for an Orco subunit.
What about you, Ali? Are you expecting that your molecules are activating odorant receptors and gustatory receptor or the receptors at the same time that we don't even know, because this physiology that we're bringing about now is something very new, to put a chemical there and to kill the eggs, basically. So there might be multiple routes of these compounds functioning with different receptors, right? So what's your thought on that?
So we already did some preliminary experiment with calcium imaging in mosquito antenna, and we found that ethyl anthranilate, which is the attractant that attract oviposition of Anopheles mosquitoes, it actually activates odorant receptors on the mosquito antenna. So we know that it activates odorant receptors, so it works through olfaction, but we don't know if it also works as a contact stimulant for oviposition. Maybe it also activates GRs as well, but we don't know yet. Yeah.
Chloe, you have been involved in this olfaction system. So do you expect that your research is going to evolve in some direction to look at the olfactory receptors or to look at the brain and how these chemicals are activated? Do you have envision in the future going in this direction, or is not something that you feel is most important for you at this point?
Yeah, that would be super interesting. One thing that we are hoping to do right now is to see for how long the effects of the soaps are lasting on the host skin. So this is one of the directions. So it's it's still at the behavioral level for the mosquitoes. But yeah, if we could, go and check, their antennae and even deeper in their brain, see how they kind of interpret this, odorant messages, that would be awesome. So yeah, maybe in the future, but for now we are trying really to understand how long those effects are lasting.
Nicoletta, you sitting there listening to this conversation, and now we are advancing the field so much, and you might feel, sorry for the lack of a better word, you might be envy because you work with ticks, and we don't know much about anything of this nature in ticks. Don't you feel like that?
Yeah, that's why I want to study ticks, and I want to try to explain more their chemiosensory system. So one thing that we want to focus particularly now, and was from thanks to the grant that I got, we want to see how the effect of climate change, especially temperature and humidity fluctuation, will impact the tick ability to detect repellent and attractant. And since ticks are very much sensitive to temperature and humidity, they like cool environment, they like humid condition. We are expecting also that this will change their sensory capability, and as a consequence, also the response to repellents and the way how we design repellents that can protect us against their bites. So it's a big open questions that we want to answer, and also looking at the pathogen impact, how this can manipulate the ability of ticks to be more resistant, for example, to extreme environmental conditions.
Joel, you had a groundbreaking discovery of the Orco receptor structure, and everyone was waiting. Joel is going to come into something new with the receptor, the odorant receptors, and all of a sudden you surprise the field and say, no, I'm working on test receptor. Did you make a pact with Josefina de Marmol not to work on receptors? Or are you still work on receptors, odorant receptors, I mean?
No packs, but I think GRs are pretty interesting. I think they sense a lot of interesting compounds. I just thought it would be a kind of a neat way to expand, I guess, what we look at, and still learn some of the same kind of basic principles. But olfactory receptors are pretty interesting too. Like you, I'm also waiting for a structure of a quote-unquote real olfactory receptor, an Orco-OR heteromer, hopefully soon.
Yes, we hear some hints, but let's not go in there. All right, so let me ask this question because we are going to have people watching from all parts of the world, and they might be looking for opportunities. So I'm going to ask this question. Are there opportunities in your lab for people to apply from outside? Sometimes I get many applications of students from overseas to study, to be PhD students in my program. And they don't understand that at the university, we are the last one to say something about the students. They have to apply. There's an independent group that run all these applications and they make a decision. And then this group will say, well, we have these students here that are eligible. Would you be interested in taking these students? I cannot before these groups, and I like this process because it's a neutral process. They make the decision, they tell us, then they come to all professors in the same field and say, we have these students available. Are you interested in being a supervisor of one of those students? So if they apply directly to me, I always take the time to try to explain it, but it's very exhausting to explain every single application. You have to say the same thing. I wish I had a default template that they could copy and paste because it's many applications that we get because the students from overseas do not understand. So having said that, I ask you, and I start with Clloé first, whether there are opportunities in your lab for applications for students and maybe postdocs and so on. Clloé, what are the opportunities in chemical ecology in your group?
Yep, we have like grad student positions. So the way it works for Virginia Tech is you need to put an application by December 15th. So so basically that would be December 15th of 2024 for fall 2025. So it's like you need to really think about it like pretty like long time ahead. But yeah, feel free to get in touch. We have several projects on mosquito plant interaction, mosquito frog interactions. So yeah, feel free to reach out. We do direct recruit, or we also go through like a lot of like screening before. So yeah, don't hesitate to reach out if you're interested in this project.
What about Acadia University? Nicoletta, in your group, any opportunities there?
Yes, we have with this grant a postdoctoral position was filled, but I have a master position to study the effect of the climate on ticks. At Acadia, basically, you can apply directly to the supervisor, and then it will be a screening through the Research and Graduate Studies Office, looking at credential if the students meet the requirement, and if it's an international student, there is also support for a visa application and so on. But I would say people that are interested, they can email me, and I can guide you through the process. I'm also the chemistry graduate coordinator, so I'm the one if the students decided to do in chemistry the master, or can be done also in biology. But yeah, we have these opportunities open.
And I see via Twitter that your students are doing very well. So can you highlight one accomplishment of your students recently?
Yes, so I have actually 3 students that graduates in their undergrads. And these students actually work all on ticks. I have my student Caris, co-supervised with Dr. Hillier. We look at hydrocarbon on ticks and how they can be involved in chemical detection and interaction. My student Claire, she looked at the octopamine and the effect, try basically to explain how essential oil works on ticks as a caricidal. So we will try to look at the mechanism. Also co-supervised with Dr. Hillier in biology. And then I have my chemistry student, Adrienne, that she did some encapsulation of thymol to develop a new caricidal natural product. And also recently, my master student, Emily, but she worked on psilocybin. So she did some encapsulation and formulation development for the delivery of psilocybin and CBD. So no chemical ecology, maybe human ecology, if we want to talk about it. Yeah.
If you are talking about training students, before Greg tells anything about Middlebury University. You have trained a very special person with a family name of Roloffs. You didn't train Wendell Roloff, did you?
No, but Wendell Roloff's granddaughter actually just graduated from Middlebury, working on the effects of road salt pollution, road salt runoff on a lot of kind of little macroinvertebrates and streams, a lot of caddisflies, mayflies, and stoneflies. And I asked her, are you related to Wendell Roloffs? And she was like, yeah, he's my grandfather. And she had no idea that he was such a big deal in insect chemical ecology. And then I got to, speak for about 10 minutes to her about all the things. And apparently it sparked a new conversation thread between them. So that's been fun.
Now tell me, are there opportunities at Middlebury and your lab in particular for students and others? You also take very good care of your students.
Thank you. Yes. So we only have opportunities for undergraduate researchers. So you have to enroll in Middlebury College first, and then you can come in. I'm hoping to get a funded postdoc soon for a project on butterfly olfaction, but we're still not there yet. So no graduate program here, just undergraduates and a potential postdoc in the future.
Ali, are you at full capacity at Drexel, or are you accepting? Let me tell you one story. That I'm going to tell this story when I give the lecture about Gerald Mywald. Once Gerald Mywald was with me, talked with me, and the postdoc from my lab came to him and asked, Jerry, do you have a postdoctoral position in your lab? And Jerry said, no, we don't have any funds. And he started telling a story. It's not for me. I'm looking for a colleague of mine, and he can do this thing, that thing, and that thing. And Jerry looked at him that, in that case, we have money. So it all depends on the candidates, right? Sometimes we don't have money, but if it's a good candidate, we might have money for that, right? But tell me a little bit about your opportunities at the Drexel, Ali.
So yeah, unfortunately, we don't have any open positions right now because we're on full capacity. I mean, we are a small lab that just started, so we're still building things. So like the electrophysiology setup, for example. So there are open projects that will open or like really start soon, and then we will need more people. But until then, I think we don't have any open positions. Drexel works as the same also with the rotation system. So people really apply for the graduate school, and then they rotate in different labs and choose which lab they join. So whenever we are open for positions, they will have to go through the Drexel system and apply for these graduate positions.
Thank you very much. Joel, what about the opportunities in your lab at Yale?
Like most of the places here, to get into Yale, into my lab as a graduate student, you have to apply to the umbrella BBS program, which I encourage everybody to do if they're interested. But we always have some space for incoming graduate students, postdocs. We're a small lab, so there's not a ton of space, but good people are always welcome to contact me if they're interested in, the structural biology of a lot of these receptors.
Okay, this is the closing now. Do you guys want to say something at the end? Just a few words or something? So thank you for being here or something like that.
I am.
Okay. All right. So thank you all. 1, 2, 3. Thank you all for being here. That was a great opportunity to hear about your progress. Now, I promise you, Wendell Roth, John Hildebrand, and Alvin He, that we would have a progress report, and here's the progress report, and it was very exciting. Thank you, Clloé.
Thank you so much.
Nicoletta, it was great to have you here.
Thanks so much, Walter, and nice to see everybody and learning more of what has been up to for us.
Greg, as always, very nice having you here.
Yes, thanks, Walter, and great to see everybody here. And it's always nice to see familiar faces when new papers get published and new press releases come out. So I'm excited to see what comes in the future.
Ali, it was wonderful having you here and hearing about the great progress that you did in the last 20 months.
Thanks, Walter. And yeah, it was really great seeing everyone. And I hope we have the opportunity to see each other in person very soon.
Joel, wonderful reading your paper and now hearing the story here in a direct conversation. Thank you for participating today.
Well, thank you very much, Walter, for bringing us back together. It's been a lot of fun. It's great to see everybody's progress and look forward to what everybody else has up their sleeves in the next year or so.
Thank you. Thank you very much, everyone, for watching. And I hope I'll see you in the Czech Republic in Prague, July 14 to 18. Again, this is the QR code that you can scan. And I'm going to put a link also underneath so that you can go and get access to the information for the annual meet of the International Society of Chemical Ecology. I'm not going to break any secrets in here, but it's a wonderful meeting because it's a family gathering. People get together and they discuss because it's not a huge meeting. Sometimes we go to meetings and if one weekend we cannot find one single person that we have been looking for, that's not the case of the meetings of the International Society of Chemical Ecology. Thank you all for the participation and for sharing such good news. It made my day, and I look forward to see you all in Prague. And another day, we might have another progress report when everyone gets tenure, because these people are in the right track to get tenure. And I'm here waiting to write letters of recommendation. Anyhow, thank you very much, everyone. It was great.
Thank you. Thank
I think
you.
it's going to be very good material. Bye-bye now.
Great.
Bye.
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 Bye.